right? 40 to 46,000 genes per minute. Pretty fast. 46,000 base pairs per minute. Sorry, not genes per minute. Base pairs per minute. How many genes does E. coli even have? We said we had 20,000, right? So how many do they have? They have about 6,000. So we have 3 times 10 to the 8 base pairs, 3 billion base pairs in our genome. And they have 4.5 million roughly in their genome. We have 20,000 and they have 6,000. So if you do like 6,000 divided by, right, and 3 million divided by 20,000, you find out, well, our genes must be huge compared to their genes, but they're not that much huger. So we just have more junk in our genome. So here again for E. coli, this shows you some of the actual real genes in E. coli and their relative location to each other. On that 100-minute map. Threonine A got the lucky gene 0. It was the beginning gene. And then everything else is mapped out relative to it. And we'll meet the lac gene. It has 3 separate genes in it. Z, Y, and A is the order they're actually in, but they list them alphabetically. We'll meet that one later in the semester. So you'll know where it is. It's about 8 minutes on the genome of E. coli. Okay? So how about those other ways? Transduction is when bacteriophage infect a cell and grab a little bit of the cell's DNA. So viruses of bacteria are called phage or bacteriophage, but they're just a virus. Like any virus, it's only metabolically active, can only do things when it's in a host cell. So the bacteriophage are going to infect, and they have DNA as their genetic material, but also some of them have RNA as their genetic material. They get that genetic material into the cell of the bacterium, and then they get more of themselves replicated. They get more of them made. They can do this with 2 sorts of strategies, or we call it a life cycle. Right? And you're like, but they're not alive. So how do you have? Yeah, the whole are viruses alive or not alive argument is just stupid. It doesn't really matter. They're viruses, okay? Like I'm saying, is a tomato a fruit or a vegetable? It's a fruit, yes, of course. There is some rules to the world, but viruses, you don't have to worry about them being alive or not alive. Just they're viruses. So one of the modes that they have is to just infect the cell and immediately begin replicating their viral particles, having their own genomes made, their own proteins made, the coats built, and they begin to process and develop, and then they get out of the cell. And in doing so, they burst their way out of the cell. They destroy the cell from the inside out, lysing the cell, L-Y-S-I-N-G, lysing, which means to burst apart. And that's known as a lytic phage. Lytic phage. It infects, replicates, destroys, moves on to the next cell. The other type of phage are called lysogenic. They enter the cell, they incorporate into the genome of the cell without replicating viral particles. They achieve replication through being part of the host chromosome, which replicates. So every time a new cell is made, one new virus genome is made, okay? At some point, though, how do they get to infect another cell? So at some point, they will be triggered to be excised from the host genome and they then go and be lytic. They do just what the lytic phages do. So a lysogenic means lysis generator. They can become lytic, but they hide out for a while without being lytic. These are sometimes known also as temperate phages. Temperate, which means they temper their response. They become lysogens first, and they only go lytic under certain conditions. And a lot of those phages were called T phages. T stands for temperate. There's T1 through 11. So here's a typical what a phage, bacteriophage will typically look like. They don't all look like this, but a typical DNA phage will look like this. All the T phages look like this, and lambda phage looks like this. They have a head which has sort of the icosahedral shape, and it's made from proteins. So of course it's not a smooth thing that doesn't look like a little spaceship kind of, but this is the proteins that make this. This is just a drawing of it. And inside here, you'll find the DNA genome. And then they have a tail. The tail has a shaft region and fibers. And it's these fibers that mediate specific interactions to proteins on the host cell. So there is cell type specificity. So not all viruses can infect, or phage could infect E. coli, for example. But a lot of them can, and they're going to do it by recognizing different proteins on the surface of the E. coli. So the E. coli could be immune to the viral infection by having defects in any of those proteins. And that's what we saw in that they had the TON mutation. It has a mutation in the protein that prevents the virus from infecting it in the first place. So no infection, no infection. But the tail protein fibers mediate that interaction. And there's a half a dozen different proteins that make up these components. So it infects the cell by attaching. Then its DNA is transferred into the host cell. And at this point, it makes a choice. Do I replicate that DNA or RNA and make more viruses, or do I integrate into the host chromosome? Now, for bacteria, only the DNA viruses can integrate into the host chromosome. So the RNA viruses are only lytic. But the DNA viruses can go either direction. So if it goes in the lytic phase, it immediately starts making new copies of its genome and transcribing that genome to make RNA and make proteins, and then make more virus proteins and make more virus particles. And then they also make enzymes that degrade the interior structures of the bacteria so that the bacteria basically decomposes from the inside out, ruptures, and the viral particles are released. Okay? A common misconception is that the cell bursts because there are so many viruses inside of it. That is not true, because A, that would be a tremendous number of viruses can fit inside of a bacterium. They don't, this happens before there would be even close to the amount of time to make that many viruses. And this is a time-consuming process. So in typical cases, this whole life cycle takes about an hour for lysis to occur with one cell. Okay. So you have the infection phase takes about 15 minutes, and then you've got to replicate, and that goes on for maybe 15 or 20 minutes, and then the lysis process begins, and eventually the cell bursts. They make specific enzymes that destroy the cell, and if those enzymes are defective, they don't ever destroy the cell. They have a hard time lysing the cell. Okay. Now, the other option available to this is to have its genome incorporate into the host chromosome creating what is known as a prophage, also known as a lysogen. Its original name, prophage, means it's going to be, it could be a phage, it's not right now. We use this terminology with viruses of eukaryotic cells like people. We have provirus, the virus which has integrated itself into the host chromosome. Like in the case of many of our retroviruses, they integrate into the host chromosome. All right, so once integrated, every time this cell replicates, makes a new cell, you make a new virus. It makes a new virus without realizing it. Except it's only making the genome of the virus. It's not making any protein. So none of the virus genes are active right now. So why go active? Why not just go like this forever? Well, eventually something generally happens to that host. That host is going to continue to grow. What's going to happen with its environment? It's probably going to stretch its resources and start running out of things and become stressed, right? And or the bacteria could be exposed to some damaging agent like UV radiation, which is bad for them. That damages their DNA. Or maybe they get attacked by another bacteria phage in the meantime, right? A different species of phage. And now that they're dealing with that. Any of these things put the bacterium under a stress response. And when it enters that stress response, there are going to be certain proteins made, and those proteins are detected by some of the bacteriophage genes. They can interact with the bacteriophage genes, and they cause the bacteriophage to be excised from the host chromosome. And once it's gone in and come back out, it won't go back in again. So it immediately goes to the lytic phage. I call this rats off a sinking ship, okay? It's perfectly happy to be on that ship as long as that ship is sailing and there's food and the people aren't finding them and they've got all the whatever they can eat on there. As soon as that ship catches on fire, those rats are like, "Nah, I'm out of here," right? As soon as there's a stress response, they're bailing. Not bailing if it's sinking. They're leaving, bailing. So off it goes. Why? Well, it has a much better chance of surviving as a bacteriophage where it can lie dormant for eons until another suitable host comes along to infect. Okay, so the little bacterial phage particles are extremely durable. In fact, the way we purify them, when we want to purify them, we use the ones that can't burst the cell. We let them grow, and they make a whole bunch in there, and then we burst the cell for them, usually using a chemical like chloroform, which chloroform is bad for cells, okay? It bursts them apart, but it doesn't do anything to the bacteriophage. In fact, we store them in the frickin' chloroform because that means nothing else can get in there with them. So that viral particle is very, very tenacious, and it's going to survive without the bacteria. It just won't make more copies of itself. But later, a new bacteria comes along, and voila, it can infect them. Okay, so those are its life cycles. What happens if, when it gets incorporated and then pops back out, what happens if it doesn't do that precisely and it accidentally takes along with it a little chunk of the bacteria chromosome? That's how transduction happens. So when this bacteriophage excises, it will do so sometimes imprecisely, and you will get a piece of the bacteria genome into the virus, and then when that virus infects another cell and goes lysogenic in the other cell, now you've transferred a bacteria gene from one bacterium to another. And that can be from one species to another. So for example, E. coli and Salmonella are very closely related bacteria, and they're both able to be infected by almost all the same bacteriophages. And if you look at their genes, a lot of their genes in E. coli are much more similar to salmonella genes and vice versa. So it's estimated that a good 10% of their genomes have actually been swapped by some sort of method, maybe like this. Because neither one of them take up DNA from the environment very easily. So it wasn't like they just got it accidentally. They probably got it from being infected with bacteriophage. Both of them are readily infected by a lot of bacteriophage. So how do bacteriophage grow? You put a whole bunch of bacteria on a petri dish, and then you mix some bacteriophage and add it on top. And the bacteriophage will infect the cell, and then it lyses that cell, moves on to the next, lyses them, moves on to the next. So it creates a little clearing known as a plaque. And you'll notice that that plaque is spelt with a Q. And a very frequent misspelling I see, especially in one of our local university publications, is that people were awarded a plaque for something, and they spell it with a Q. I'm like, yay, bacteriophage. Plaque, like a plaque, is with a G. So anyway, there you go. We'll talk a little bit more about bacterial genes on next week, and then we'll have a quiz on Tuesday. So don't pass that on.